the invitation to talk this afternoon, Thomas. It's much appreciated. And what I want to tell people is a smaller part of a much bigger story. So I've had a postdoc in my lab, James, for a few years now working on a BPSRC grant. And I just want to tell you one bit of his work, which is trying to develop an assay using time-resolved FRET to explore inhibitor and substrate binding to the multi-drug pump ABCG2. As we've heard, ABCG2 is a broad substrate specificity transporter. It's localized to many so-called sanctuary sites in the body. In terms of cancer drug resistance, we know it is clinically relevant, especially in leukemia, but there are some solid tumors where expression of ABCG2 is a negative prognostic indicator. We know also that due to its localization in things like the gut and the kidney, that it affects the pharmacokinetic profiles of many drugs. And slightly different to ABCG1 that we've heard quite a bit about, we know that G2 has to function as a dimer because it's a so-called half transporter. So in terms of drug binding and transport, we know that G2 is polyspecific, and I've just listed here a handful of chemicals or types of chemicals that we know that ABCG2 can interact with. And Anna knows probably thousands more that it can interact with and how. Those range from physiological substrates, which include urate and probably include some heme-based compounds, to clearly non-physiological pharmacotherapeutic compounds, such as statins and anti-cancer drugs. And we know, as Michael has said earlier, that there's some overlap with the substrate repertoire of another multi-drug pump, PGP, ABCG1. And we're interested in this polyspecificity. And there are many ways in which people have investigated this. And these includes looking at the transport mediated by ABCG2, either in cells or in inside-out vesicles, or measuring the transport across polarized cells, or looking at the ability to stimulate the ATPase assay, to stimulate the ATPase activity in membrane preparations, or to just see if you can solve the crystal or the cryo-EM structure with your favorite drug bound to the protein. And there's been a lot of advances in that for ABCG2. And then there's also a whole range of mutational analysis. Can we somehow perturb the pump and show that that affects drug binding and transport? So one of our aims in this grant was to see if we could develop a technique that was purely focused on the first step, translocation. In other words, could we get a technique working that looked at how drugs bind, interact with the transporter? And the assay we've been working on is a FRET-based assay, which is shown schematically on this slide. So what you're looking at this slide, hopefully my pointer is showing for you, is in both cases we have one of the ABCG2 structures. And on the left-hand side, we have this orange circle. This is a fluorescent molecule bound in ABCG2's so-called cavity 1. And this fluorophore is accepting resonance energy from a terbium atom. And that resonance energy transfer can be detected to show that that a fluorescent molecule binds to ABCG2. And I'll explain more how we get the terbium atom near our fluorescent compound in a minute. On the right-hand side, we show what happens if you have the exact same setup, but in addition to this fluorescent compound that binds to G2, you've also had another non-fluorescent compound that would displace this molecule. And in that case, we no longer get a FRET between the terbium and the fluorescent ABCG2 substrate or inhibitor. So this is the principle of our assay, to use the light emitted from excitation of terbium atoms to measure fluorescence binding at ABC2. And the way we've tagged our protein with terbium is to take advantage of a protein tag known as the SNAP tag. Now, the SNAP tag is a smallish 18 kilodalton protein. It's an alkyl guanine DNA alkyl transferase protein. And this means it can bind covalently with anything that is that has an O6 alkyl guanine. And so we've tagged ABCG2 with this so-called SNAP tag. This protein is then labeled with an Oregon green version of O6 alkyl guanine, and that lets us show that our SNAP tagged ABCG2 is present at the cell surface. On the right-hand side of this slide, we can also show that this SNAP tag G2 is still functional. So this is a conventional transport assay looking at the accumulation of mitoxantrone. It's probably just worthwhile looking at the bottom half of this panel. So when we add mitoxantrone to cells expressing this SNAP tagged ABCG2, then they do not accumulate much mitoxantrone. That's this population, this fluorescence of mitoxantrone on this axis. When we add the classic inhibitor of ABCG2, which is KO143, we shift this population to the right, which shows that they're now accumulating mitoxantrone. So this SNAP tagged version of the transporter is at the cell surface and it's functional in a transport assay. And what we then designed and made was a version of KO143 bound to a BODIPI fluorophore. So KO143 is a specific inhibitor, or reasonably specific inhibitor, to be correct for Michael's benefit, of ABCG2. And when we add this BODIPI fluorophore to it, it still retains that inhibition, which is shown on the right-hand side of this slide. We then undertook this TR-FRET assay using this BODIPI BY630 KO143, so this fluorescent inhibitor of ABCG2. And we could show specific and saturable binding of BODIPI KO143 to ABCG2 with an affinity of about 50 nanomolar, but which is very, very similar to the sort of concentrations of KO143 that inhibit ABCG2 in cell and membrane assays. I should say all of these assays are done in cell membrane. We have extended this. We've got a whole range of mutants of ABCG2 where we've made changes in the drug binding pocket, changes in the pocket that should interfere with KO143 binding. And that data is not quite finished. That's why it's not on this slide, but it does seem to show that where we mutate residues that should interact with KO143, we do perturb this saturation binding isotherm, and we do see changes in affinity. What we've also done is we've shown that this fluorescent inhibitor, it does get displaced, so it doesn't just bind to G2 and sit there. It can be displaced in a sort of homologous fashion by KO143. So this is now the FRET ratio plotted against the concentration of the displacing ligand, and you can see that KO143 can displace its fluorescent counterpart with an IC50, again in a sensible range for this inhibitor, about 100 nanomolar. So we can use this fluorescent KO143 and this TR-FRET assay to measure specific binding, and we can measure competition as well with a high affinity G2 inhibitor. We can also start to measure substrate interaction with the transporter by looking at substrate's ability to displace the BODIPI KO143. We've looked at 3 substrates. I'm going to just zoom my screen a little bit because otherwise I can't see my own data, to show here that we can displace our fluorescent KO143 both with estrone sulfate and with imatinib. We can't displace it with a couple of compounds that we don't think G2 interacts with. There's no evidence in the literature that either aspirin or allopurinol interact with ABCG2, and we don't find that they're able to displace our fluorescent KO143, which affords us some confidence. What we are seeing is an ABCG2 specific phenomenon. And so I'm happy to wrap up there and say that this TR-FRET assay looks as though it allows us to measure binding as opposed to transport at ABCG2, and allows us to work with both wild type and mutant proteins and start to look at a wider range of substrates and other ABCG2 inhibitors. I think I've kept pretty much the time there, and I'm happy to answer a couple of questions if there are any.
Thanks a lot here. Very interesting and very clear presentation, and to the point and to the time. So it makes a lot of sense to do this FRET assay. How large is the terbium atom? I mean, does it interfere with the structure of ABCG2 or?
I mean, it shouldn't do. The snap tag is at the N terminus of ABCG2. There is a short linker before the rest of the MBD. And so what we don't know is exactly where that snap tag is spatially. But it's hanging off the end of the MBD. It shouldn't be interfering with anything going on in the membrane.
Right. And do you plan to make any ATPase mutants also?
So we have got the classic Walker B mutation E211Q in ABCG2, and we have the data for that is predicted. Sorry, our prediction from that is that it would shift this curve. It would produce a shift in the affinity. It would reduce the affinity, I suspect. We haven't done the experiments yet, Thomas. One of the downsides of this is that the chemists were unable to make a massive amount of this compound for us. And so we've had this awkward task of prioritizing the experiments. This is one of the next ones to do.
Right. Okay. All sounds good. Good luck. Oh, I have a question for Michael.
Yes. In a very interesting and really very nice assay. One of the peculiarities of P-glycoprotein is that substrate the residues in the substrate binding domains that should be interacting with substrates, you can mutate them and not see very dramatic effects on transport. It's almost as if there are primary and secondary and tertiary and quaternary binding sites. So you can substitute lots of alanines in the substrate binding site. This is work that Suresh Ambudkar has done. Do you know if the same is true in ABCG2? Have you looked at your mutants which affect substrate binding by your assay to determine whether they actually are affecting transport?
So one of the things I think literature tells us is that most of the multi-drug pumps have the potential to make lots and lots of weak but accumulating interactions with their transport substrates. And so that single point mutations may sometimes not have a big impact because it's another residue nearby that sort of then mops up and vowels interaction. So I think single point mutations are a difficult with any assay. Some of the mutations I mentioned in passing are not single point mutations. They're multiple mutations at different points in the pocket where we hope to be able to see a bigger impact.
All right. So thanks.
May I ask a
Oh,
question?
yeah, sure. Anna, go ahead.
I've just clicked
I'm
stop
sorry,
share, but I'll ask the question.
I would like to ask you for the conditions you measured I mean, did you have, what is the ATP and the magnesium concentration?
So this is all done in membranes that we anticipate are ATP free, Anna. So one of the things we want to do is also run this with AMP, PNP, and trap transporter in that ATP bound state.
This would be very interesting because this is probably the assumption that you have an open conformation when you have no ATP, and then you have the binding in this open A shape, and you have probably, so it would be very
Absolutely.
interesting to do it in the presence of nucleotides.
Yeah, we're very excited to do those experiments. Yes. Like
very
some of
interesting.
the data on here was obtained yesterday, so we're quite. If you call me in a week's time, we might have some of that data.
Very good. I'll call you. Thank you very much.
So thanks again.